Dear students, a warm welcome to the class on oogenesis. Oogenesis is a type of gametogenesis. Gameto means gametes and genesis means formation. Gametogenesis is the process of formation of gametes. There are two types of gametes: sperm, the male gamete, and ovum, the female gamete. Gametogenesis is of two types: spermatogenesis and oogenesis. Sperma in Greek means seed or germ, which is also related to sperm, and genesis is formation. So spermatogenesis is the process of formation of sperm. Oogenesis. Oon means egg or ovum, and genesis means formation. So oogenesis is the process of formation of ovum. In today's class, we will be studying in detail about oogenesis. By definition, oogenesis is the process of formation of ovum from oogonia in the ovary. Suppose this is the ovary, the female gonad. The ovum is formed from its mother cell, the oogonia. The process of formation of ovum from oogonia in the ovary is called as oogenesis. Oogonia is diploid in number and ovum is haploid in number. Remember, all the cells of our body are diploid in number except for the gametes. Oogenesis starts during the fetal life. Before moving on to oogenesis, we will study the structure of ovary. And all the terms that are highlighted or presented in the slide are important to study the process of oogenesis. Ovary is a solid mass. Any solid structure in our body has two parts. The solid mass is divided into outer cortex and inner medulla. Now this outer cortex here is lined by cells called as the germinal epithelial cells. The germinal epithelial cells of the ovary multiply to form oogonia, which is the mother cell of the ovum, which further grows to primary oocyte, which then forms secondary oocyte, and this develops into a mature ovum. This entire process we'll be studying during the process of oogenesis. The cortex region over here has some circular structures. These structures are called as follicles. So there are some primitive follicles over here which are called as primordial follicles. These follicles will develop into primary follicle. Primary will develop into secondary follicle. Secondary will develop into tertiary follicle. Tertiary will develop into mature follicle, which is called as Graafian follicle. And there are some other structures over here which are called as corpus luteum and corpus albicans, which you have already studied in menstrual cycle. Now here, this portion is the medulla, which has blood vessels in it. Moving on to the development of follicles. The first follicle we are studying is the primary follicle. Now, the primary follicle has primary oocyte in it. This is the primary oocyte, which is surrounded by a layer of granulosa cells. These granulosa cells provide nourishment to the primary oocyte, and this is the diffusing membrane, that is the zona pellucida. So a primary follicle is made up of primary oocyte and it is surrounded by granulosa cells. The primary oocyte is formed from oogonia. Oogonia is the mother cell of ovum. Now, the primary follicles are formed during the fetal life itself, and they're formed at about 3rd to 4th month of fetal life. These cells proliferate from 3rd to 5th month in large amounts and reach millions. Later, by the end of the fetal life, they start degenerating, and this degeneration process continues even after birth to puberty. At puberty, around 60,000 to 80,000 primary follicles are left in each ovary. I'll once again repeat primary follicle. So primary follicle as a primary oocyte as granulosa cells to provide nutrition. They increase in number during the fetal life, but from the end of the fetal life to puberty, they start degenerating, and at the puberty, there are around 60,000 to 80,000 primary follicle left in each ovary. No more primary follicles are added after birth. The primary follicle now develops into a secondary follicle. You can see the secondary follicle is larger than the primary follicle. The difference here is it has few more layers of granulosa cells, and it has a new layer of cells here, that is the theca cells. The primary oocyte is also present in the secondary follicle. So secondary follicle consists of a primary oocyte, it has granulosa cells and theca cells. The secondary follicle will now develop into a tertiary follicle. The tertiary follicle, if you can see here, it has a fluid-filled cavity that is called as antrum, which is developed in between the granulosa cells. And you can see more layers of granulosa cells over here. One more important feature here is the theca layer. It is differentiated into inner theca interna and outer theca externa. So the theca layer here is differentiated. One more important change here is the primary oocyte that was present in the primary follicle and the secondary follicle is now changing to secondary oocyte. I'm sorry, the primary oocyte that was present in the primary follicle and the secondary follicle is now becoming secondary oocyte. The tertiary follicle now grows into a Graafian follicle. The Graafian follicle is a mature follicle. You can see the size of the antrum here. It is increased in size. The granulosa cells are present. There is theca interna, theca externa. There is a stalk-like structure over here, which is present near the secondary oocyte. It is called as cumulus oophorus. The most important feature that is taking place, or most important event that is taking place in the Graafian follicle is ovulation. Now, ovulation takes place on the midday of the menstrual cycle. In human beings, the menstrual cycle biologically is a 28-day cycle, and the midday is the 14th day. On the 14th day, the Graafian follicle ruptures to release the secondary oocyte. So this process is called as ovulation, and this is an important event that is taking place in this follicle. So these are the 4 types of follicles. We have learned about the primary follicle, secondary follicle, tertiary follicle, and Graafian follicle. Let's quickly see the structure again. The germinal epithelium multiplies and forms oogonia. Millions of oogonias are formed, and further they form primary oocyte. Primary oocytes are also millions in number, which further degenerate from fetal life to puberty. Here what you can see, the circular structure is the primary oocyte. The primary oocyte is surrounded by granulosa cells and it is called as primary follicle. The secondary follicle as granulosa cells, it has theca cells and primary oocyte, it is the secondary follicle. The tertiary follicle here has granulosa cells, it has theca interna, theca externa, it has antrum, and the change here is it has secondary oocyte. Next is the Graafian follicle, which is almost having the similar structures as that of the tertiary follicle, but only change here is the cumulus oophorus. Here the Graafian follicle, you can see, ruptures on the midday of the menstrual cycle, releasing its secondary oocyte, and this process is called as ovulation. the Graafian follicle ruptures, releasing the secondary oocyte, and this process is called as ovulation. Later, the leftover parts of the Graafian follicle forms corpus luteum. Corpus luteum is important over here because it secretes a hormone called as progesterone, which is a pregnancy hormone which helps in maintaining pregnancy. If there is no pregnancy, then the corpus luteum forms corpus albicans. So this is about the structure of ovary. We have studied about the development of follicles. Now we will study the process of oogenesis. As we all know, oogenesis is the process of formation of ovum from oogonia. Oogonia is the mother cell of the ovum. Oogonia undergoes mitosis to form more oogonias. Mitosis is a equational division. During mitosis, the mother cell divides to form two daughter cells which are of same size and shape. You can see here the oogonia is diploid in number. In humans, the chromosome number is 46. And since the oogenesis takes place in females, we have the sex chromosome as X chromosomes here. The 44 are the pair of autosomes. 44 is the number of autosomes and two sex chromosomes. So 44, 45, 46. So when this oogonia undergoes mitosis, two daughter cells are formed with same number of chromosomes. I hope this is clear. This continues further. These oogonias continue multiplication to form more oogonias. You can see here one cell has formed two cells has formed four cells, four will form 8 to 16, and this way it forms millions of oogonias in the fetal ovary. This phase is called as multiplication phase, and this phase takes place in the fetal ovary before birth. Now, some of the oogonias, not all the oogonias, some of the oogonias will develop into primary oocyte. Some of the oogonias, here there are four oogonias, we have just taken one of it. Okay, so out of the four oogonias or out of the many oogonias, few develop into primary oocyte. This is also taking place in the fetal life. The primary oocyte also has the same number of chromosomes because there is no multiplication over here. Only the certain oogonias have increased in the amount of cytoplasm and they have increased in size. So that is why this is called as growth phase. Now, the primary oocyte undergoes meiosis 1 and gets arrested at prophase 1. The primary oocyte undergoes meiosis 1 and it gets arrested at prophase 1. This is also taking place during the fetal life. The primary oocyte, what is meiosis? We have already studied about mitosis, but what is meiosis? Meiosis is a reductional division. Henceforth, we will see the 46 chromosomes being reducing to 23. Meiosis has two phases, meiosis 1 and meiosis 2. In turn, meiosis 1 as prophase 1, metaphase 1, anaphase 1, telophase 1. Similarly, meiosis 2 also has prophase 2, metaphase 2, anaphase 2, and telophase 2. This is taking place, this arresting in meiosis prophase 1 is taking place in the fetal life itself, and here the primary oocyte is frozen in this particular phase from birth to puberty. I'll repeat this once again. The primary oocyte starts meiosis 1 and gets arrested at prophase 1 in the fetal life itself. And it continues to be in this phase till puberty. That's the reason oogenesis is called as a discontinuous process. That is, it takes place during the fetal life, and then there is a gap from birth to puberty, and again it resumes during puberty. I hope this is clear. Now here if you see, the primary oocyte in the fetal life gets surrounded by granulosa cells and forms primary follicle, which we have already studied. The primary oocyte gets surrounded by a layer of granulosa cells and it forms primary follicle. It remains the same from birth to puberty. Now at puberty, now this process is from puberty to menopause. At puberty, the a group of primary oocytes, you know now there are 60,000 to 80,000 primary oocytes left in each ovary. A group of primary oocytes, say 15 to 20 primary oocyte, starts continuing or starts resuming its meiosis, but ultimately only one will proceed its division. The primary oocyte that was arrested at the prophase 1 completes its meiosis 1 in the tertiary follicle. So it completes its meiosis 1 in this follicle, that is the tertiary follicle, to form secondary oocyte and first polar body. The secondary oocyte and first polar body, if you can see here, they are different in size. The size of the secondary oocyte is larger because it has retained the cytoplasm from the primary oocyte, and the first polar body has less amount of cytoplasm. Therefore, the first polar body will degenerate. Even if it multiplies further, it is not functional, it will degenerate. Now you can also see the chromosome number over here. The chromosome number is reduced to half. The half of 44 is 22 is gone to this cell, 22 is gone to this cell. In this 2x chromosome, 1x goes to this cell and the other x goes to another cell. Next, the secondary oocyte starts its meiosis 2 and gets arrested at metaphase 2. The secondary oocyte here in the tertiary follicle starts its meiosis 2 and gets arrested at metaphase 2. Then on we have studied the tertiary follicle develops into Graafian follicle, and the Graafian follicle will rupture, releasing the secondary oocyte. That is this oocyte, which is still not completed its meiosis 2. It is arrested in metaphase 2. Now, the secondary oocyte completes its meiosis 2 only if it is fertilized by sperm. This secondary oocyte that is released by the Graafian follicle or in turn released by the ovary enters the fallopian tube, and in the fallopian tube, if there is sperm and if it fertilizes the secondary oocyte, then the meiosis 2 is completed, resulting in the formation of mature ovum and a second polar body. So this is the mature ovum here. The mature ovum, immediately the nucleus of the ovum fuses with the nucleus of the sperm and forms a zygote. Here the second polar body is formed. Again, you can see the difference in the size here. The cytoplasm is more in the ovum or the ootid, and the second polar body has less cytoplasm. So here this will degenerate. So this is the process of oogenesis. I'll repeat this once again. The oogonia is the mother cell of the ovum. It undergoes mitosis to form more oogonias. It further undergoes mitosis to form more and more oogonias, and it increases in number. This is called as the multiplication phase that takes place during the fetal life. Next is the growth phase, which also takes place in the fetal life. Some of the oogonias will grow in size, that is increase its cytoplasm and form primary oocyte. This primary oocyte in the fetal life itself undergoes meiosis 1 and gets arrested at prophase 1, and this will continue from birth to puberty. The primary oocyte is frozen in this stage of prophase 1 of meiosis 1 from birth to puberty. Now oogenesis resumes again at puberty and then starts the maturation phase. During At puberty, the primary oocyte completes its meiosis 1 in the tertiary follicle and forms secondary oocyte and first polar body. It is a reductional division, therefore the chromosome number is reduced to half. The secondary oocyte is larger in size and the first polar body is smaller in size because of the unequal distribution of cytoplasm. Thereby, the first polar body is non-functional, it degenerates. The secondary oocyte in the tertiary follicle starts its meiosis 2 and gets arrested at metaphase 2. Secondary oocyte starts its meiosis 2 and gets arrested at metaphase 2 and completes its meiosis 2 only if it is fertilized by the sperm. If it is fertilized by the sperm, the secondary oocyte forms a ovum and a second polar body. Again, they are of two different sizes because of the unequal distribution of cytoplasm, and second polar body degenerates. And this is the mature ovum that is formed, and this is the process of oogenesis, where a ovum is formed from oogonia. This is what is happening once the ovum is released from the ovary, that is the secondary oocyte, immature ovum, is released from the Graafian follicle, which has not completed its meiosis 2, which is arrested at metaphase 2. It enters into this is the secondary oocyte, it enters into the fallopian tube, and here you can see it is fertilized by the sperm. Only if it gets fertilized by the sperm, then the meiosis 2 is completed, resulting in the formation of a mature ovum and second polar body. Now the mature ovum, as soon as the mature ovum is formed, the nucleus of the mature ovum fuses with the nucleus of the sperm and the zygote is formed. You can see here the zygote is formed. Yes. So this process is ovulation, and this you've already seen. This is how the sperm fertilizes the secondary oocyte to form a ovum, and then only the zygote is formed. So this was the process of spermatogenesis. So this is the notes on the process of oogenesis. So oogenesis starts during fetal life. Multiplication phase. In multiplication phase, oogonia undergoes mitosis to increase in number. No more oogonias are formed after birth. In growth phase, oogonias develop into primary oocyte. The primary oocyte undergoes meiosis 1 and gets temporarily arrested at prophase 1 of meiosis 1. In maturation phase, that is at puberty, the primary oocyte within the tertiary follicle grows in size and completes its first meiotic division, forming a large haploid secondary oocyte and a tiny first polar body. The polar body degenerates. The secondary oocyte retains bulk of nutrient-rich cytoplasm of the primary oocyte, and it starts its meiosis 2 in the tertiary follicle and gets arrested at metaphase 2. The secondary oocyte completes its meiosis 2 only after fertilization with the sperm, resulting in the formation of a mature ovum and second polar body. So this is very important question for 5 marks. Next, we will talk about the differences between spermatogenesis and oogenesis. Spermatogenesis is the process of production of sperms, and oogenesis is the process of formation of ovum or egg. Next, it occurs in the seminiferous tubules of the testis, whereas oogenesis occurs in the ovary. The third difference is spermatogenesis is a continuous process, whereas oogenesis, as we already discussed, it is a discontinuous process. It starts at fetal life, then resumes at puberty. So in between, the process is frozen or it is in the resting phase. The next difference is all the spermatogonia divide to form sperms, whereas in oogenesis, only some oogonias give rise to oocytes, that is primary oocytes, whereas in spermatogenesis, all the spermatogonias divide to form spermatocytes, and these spermatocytes further form sperms. Spermatogenesis is a process that begins at puberty and continues till the end of life, whereas oogenesis starts at fetal life and it ceases at menopause. So it doesn't continue till the end of the life, it ceases at menopause. Spermatogenesis, here the cells have equal cytoplasmic division, or there is equal distribution of cytoplasm in each cells. So all the 4 sperms that are formed from a single spermatogonia are equal in size and shape. Whereas in oogenesis, the cytoplasmic division is unequal. That's the reason we see secondary spermatocytes and ovum being larger and the polar bodies being smaller. From one spermatogonia, 4 motile sperms are formed in spermatogenesis. But in oogenesis, only one ovum is formed from a oogonia. So mother cells in both the both the types of gametogenesis, there's only one mother cell. In spermatogenesis, it is spermatogonia. In oogenesis, it is oogonia. But in spermatogenesis, 4 sperms are formed, and in oogenesis, only one ovum is formed. In spermatogenesis, motile gametes are formed. The male gametes are motile. In oogenesis, we see a non-motile ovum is formed. So female gamete is non-motile. All the stages of spermatogenesis are completed in testis, whereas in case of oogenesis, the major portion of the oogenesis is in the ovary, but the last few stages you have seen it takes place in the oviduct, that is the fallopian tube. So we have finished 2 important topics here: oogenesis and differences between the spermatogenesis and oogenesis. Spermatogenesis, oogenesis and their differences are very, very important questions for your exams. These are some of the questions for your assignment. So please answer these questions, as these questions are very important. Thank you, students. If you have any doubt, reach back to us. Have a pleasant week.